A highly integrated DNA nanomachine operating in living cells, powered by an endogenous stimulus. Synthetic molecular machines have received increasing attention because of their great ability to mimic natural biological motors and create novel modes of motion. However, very few examples have been implemented with real autonomous movement inside living cells due to the challenges of the driving force and highly integrated system design. In this work, we report an elegant, highly integrated DNA nanomachine that can be powered by endogenous ATP molecules and autonomously operate inside living cells without any auxiliary additives. It assembles all components on a single gold nanoparticle, including a hairpin-locked swing arm, encoding a start triggered by an intracellular target molecule, and a two-stranded DNA track responding to the motion of the swing arm. When the intracellular target activates the nanomachine via the unlocking swing arm, the machine autonomously and progressively operates on the established DNA track via intramolecular toehold-mediated strand migration and internal ATP binding. This paper also demonstrates the machine's bioanalytical application for specific microRNA, miRNA, imaging in living cells. A vast range of natural protein motors exist in the biological world, and they participate in significant biological processes, such as mysin and muscle contraction, kinesin and dynein and cellular cargo transport, and DNA polymerase and DNA synthesis. Inspired by these biological motors, researchers have attempted to fabricate various artificial molecular machines and motors that can be precisely controlled and exploited on the nanometer scale, simultaneously connecting molecular systems to the macroscopic world. Among them, synthetic DNA-based machines with a simple DNA structure, high programmability, and predictability of Watson/Crick base pairing, good structural robustness, and low cost of synthesis, exhibit significant advances that enable the fully de novo design of diverse and powerful devices. So far, many DNA machines, such as walkers, rolling motors, tweezers, gears, springs, robots, and transporters, have been created to mimic the movement of protein motors, or to have distinct behavior patterns, and successfully tested in cell-free settings. Fabricating DNA nanodevices that could particularly operate inside living cells to execute design tasks, such as in vivo imaging, biological computing, and autonomous theranostic application, has long been a challenging goal. It is noteworthy that most of the reported sophisticated DNA machines are designed with complicated DNA origami tracks, large-scale nanomaterials, or complex motion patterns, which have usually limited their potential applications in living cells. I'm going to leave the link in the description for those who want to keep reading. But for now, let's go over here, and we're going to read DNA Nanomachines. This was released in 2007. We are learning to build synthetic molecular machinery from DNA. This research is inspired by biological systems in which individual molecules act singly and in concert as specialized machines. Our ambition is to create new technologies to perform tasks that are currently beyond our reach. Beyond our reach. Yeah, in 2007. DNA nanomachines are made by self-assembly, using techniques that rely on the sequence-specific interactions that bind complementary oligonucleotides together in a double helix. They can be activated by interactions with specific signaling molecules or by changes in their environment. Devices that change state in response to external trigger might be used for molecular sensing, intelligent drug delivery, or programmable chemical synthesis. Biological molecular motors that carry cargoes within cells have inspired the construction of rudimentary DNA walkers that run along self-assembled tracks. It has even proved possible to create DNA motors that move autonomously, obtaining energy by catalyzing the reaction of DNA or RNA fuels. Now let's go over here, and we're going to read. Graphene-based nano antennas may enable networks of tiny machines. Networks of nanometer scale machines offer exciting potential applications in medicine, industry, environmental protection, and defense. But until now, there's been one very small problem: the limited capability of nanoscale antennas fabricated from traditional metallic components. With antennas made from conventional materials like copper, communication between low-power nanomachines would be virtually impossible. But by taking advantage of the unique electronic properties of the material known as graphene, researchers now believe they are on track to connect devices powered by small amounts of scavenged energy. Based on a honeycomb network of carbon atoms, graphene could generate a type of electronic surface wave that would allow antennas just 1 micron long and 10 to 100 nanometers wide to do the work of much larger antennas. While operating graphene nano antennas have yet to be demonstrated in 2013, 13 years ago, way before COVID. The researchers say their modeling and simulations show that nano networks using the new approach are feasible with the alternative material. We are exploiting the peculiar propagation of electrons in graphene to make a very small antenna that can radiate at much lower frequencies than classical metallic antennas of the same size, said Ian Achildes, a Kent Byers Chair Professor in Telecommunications in the School of Electrical and Computing Engineering at the Georgia Institute of Technology. We believe that this is just the beginning of a new networking and communication paradigm based on the use of graphene. Sponsored by the National Science Foundation, the research is scheduled to be reported in the journal IEEE Journal of Selected Areas in Communications. In addition to the nanoscale antennas, the researchers are also working on graphene-based nanoscale transceivers and the transmission protocols that would be necessary for communication between nanomachines. The communications challenge is at the micron scale. Metallic antennas would have to operate at frequencies of hundreds of terahertz. While those frequencies might offer advantages in communication speed, their range would be limited by propagation losses to just a few micrometers. And they'd require lots of power, more power than nanomachines are likely to have. Achildes has studied nano networks since the late 1990s and had concluded that traditional electromagnetic communication between these machines might not be possible. But then, he and his PhD student, Joseph, who graduated in August 2013 and is now an assistant professor at the State University of New York at Buffalo, began reading about the amazing properties of graphene. They were especially interested in how electrons behave in single-layer sheets of the material. When electrons in graphene are excited by an incoming electromagnetic wave, for instance, they start moving back and forth, explained Achilles. Because of the unique properties of the graphene, this global oscillation of electrical charge results in a confined electromagnetic wave on the top of the graphene layer. Known technically as a surface plasmon polariton, SPP wave, the effect will allow the nano antennas to operate at the low end of the terahertz frequency range, between 0.1 and 10 terahertz, instead of 150 terahertz required by traditional copper antennas at nanoscale sizes. For transmitting, the SPP waves can be created by injecting electrons into the dielectric layer between the graphene sheet. Materials such as gold, silver, and other noble metals can also support the propagation of SPP waves, but only at much higher frequencies than graphene. Conventional materials, such as copper, don't support the wave. By allowing electromagnetic propagation at lower terahertz frequencies, the SPP waves require less power, putting them within range of what might be feasible for nano machines operated by energy harvesting technology pioneered by Ling Wang, a professor in Georgia Tech School of Materials Science and Engineering. With this antenna, we can cut the frequency by 2 orders of magnitude and cut the power needs by 4 orders of magnitude. Using this antenna, we believe that energy harvesting techniques developed by Dr. Wang would give us enough power to create a communications link between nano machines. The nano machines and the network that Achilles and Jornet envision would include several integrated components. In addition to the energy harvesting nano generators, there would be nanoscale sensing, processing, and memory, technologies that are under development by other groups. The nanoscale antenna and transceiver work being done at Georgia Tech would allow the devices to communicate the information they sense and process to the outside world. Each one of these components would have a nanoscale measurement, but in total, we would have a machine measuring a few micrometers, said Jornet. There would be lots of trade-offs in energy use and size. Beyond giving nano machines the ability to communicate, hundreds or thousands of graphene antenna transceivers, sets, might be combined to help full-size cellular phones and internet-connected laptops communicate faster. The terahertz band can boost current data rates in wireless networks by more than 2 orders of magnitude, Achilles noted. The data rates in current cellular systems are up to 1 gigabit per second in LTE advanced networks, or 10 gigabits per second in so-called millimeter wave, or 60 gigahertz systems. We expect data rates on the order of terabits per second in the terahertz band. The unique properties of graphene, Achilles says, are critical to this antenna and other future electronic devices. Graphene is a very powerful nanomaterial that will dominate our lives in the next half-century, he said. The European community will be supporting a very large consortium involving many universities and companies with an investment of 1 billion euros to conduct research into this material. Researchers have so far evaluated numerous nano antenna designs using modeling and simulation techniques in their laboratory. The next step will be to actually fabricate a graphene nano antenna and operate it using a transceiver, also based on graphene. Our project shows that the concept of graphene-based nano antennas is feasible, especially when taking into account very accurate models of electron transport in graphene, said Achildes. Many challenges remain open, but this is a first step forward, creating advanced nano machines with many applications in the biomedical, environmental, industrial, and military fields. So this article was released almost 10 years before COVID. And what did everybody take when they took the COVID shot? Now, let's go over here, and we're going to look at this PDF file. This is from the IEEE Communications Magazine, 2015. The Internet of Bio-Nanothings. The Internet of Bio-Nanothings stands as a paradigm-shifting concept for communication and network engineering, where novel challenges are faced to develop efficient and safe techniques for the exchange of information, interaction, and networking within the biochemical domain, while enabling an interface to the electrical domain of the Internet. The Internet of Things, IoT, has become an important research topic in the last decade, where things refer to interconnected machines and objects with embedded computing capabilities employed to extend the Internet to many application domains. While research and development continue for general IoT devices, there are many application domains where very tiny, concealable, non-intrusive things are needed. The properties of recently studied nanomaterials, such as graphene, have inspired the concept of Internet of Nanothings, IoNT, based on the interconnection of nanoscale devices. Despite being an enabler for many applications, the artificial nature of IoNT devices can be detrimental, where the deployment of nanothings could result in unwanted effects on health or pollution. The novel paradigm of the Internet of Bio-Nanothings is introduced in this paper by stemming from synthetic biology and nanotechnology tools that allow the engineering of biological embedded computing devices based on biological cells and their functionalities in the biochemical domain. Bio-Nanothings promise to enable applications such as intra-body sensing and actuation networks, and environmental control of toxic agents and pollution. The Internet of Bio-Nanothings stands as a paradigm-shifting concept for communication and network engineering, where novel challenges are faced to develop efficient and safe techniques for the exchange of information, interaction, and networking within the biochemical domain, while enabling an interface to the electrical domain of the Internet. The Internet of Things, IoT, defines a cyber-physical paradigm where all types of real-world physical elements, sensors, actuators, personal electronic devices, or home appliances, among others, are connected and are able to autonomously interact with each other. This new form of seamless connectivity is the enabler for many applications, such as machine-to-machine communication, real-time monitoring of industrial processes, smart cities, smart grids for energy management, intelligent transportation, environmental monitoring, infrastructure management, medical and healthcare systems, building and home automation, and large-scale deployments. The Internet of Things became a focus for research and development in the last 15 years. Again, this was released in 2015. A large amount of investments for Internet of Things was and is still being made by government agencies and industry worldwide. Recently, the concept of IoT has been revised in light of novel research advances made in the field of nanotechnology and communication engineering, which enable the development of networks of embedded computing devices based on nanomaterials such as graphene or metamaterials, having scales ranging from 1 to a few hundred nanometers, called nanothings. The Internet of Nanothings, IoT, introduced for the first time in number 1, is proposed as the basis of numerous future applications, such as in the military, healthcare, and security fields, where the nanothings, thanks to their limited size, can be easily concealed, implanted, and scattered in the environment, where they can cooperatively perform sensing, actuation, processing, and networking. While nanothings can push the engineering of devices and systems to unprecedented environments and scales, similarly to other devices, they have an artificial nature since they are based on synthesized materials, electronic circuits, and interact through electromagnetic communications. These characteristics can be detrimental for some application environments, such as inside the body or in natural ecosystems, where the deployment of nanothings and their electromagnetic radiation could result in unwanted effects on health or pollution. A novel research direction in the engineering of nanoscale devices and systems is being pursued in the field of biology by combining nanotechnology with tools from synthetic biology to control, reuse, modify, and re-engineer biological cells. By stemming from an analogy between a biological cell and a typical IoT embedded computing device, a cell can be effectively utilized as a substrate to realize a so-called bio-nano thing through the control, reuse, and re-engineering of biological cells' functionalities, such as sensing, actuation, processing, and communication. Since cells are based on biological molecules and biochemical reactions rather than electronics, the concept of Internet of Bio-Nanothings, introduced in this article, is expected to be paradigm shifting for many related disciplines, such as communication and network engineering, which is the focus of this article. The execution of DNA-based instructions, the biochemical processing of data, the transformation of chemical energy, and the exchange of information through the transmission and reception of molecules, termed molecular communication, are the basis of a plethora of applications that will be enabled by the Internet of Bio-Nanothings, such as intra-body sensing and actuation, where bio-nanothings inside the human body would collaboratively collect health-related information transmitted to an external healthcare provider through the internet, and execute commands from the same provider, such as synthesis and release of drugs. Intra-body connectivity control, where bio-nano things would repair or prevent failures in the communications between our internal organs, such as those are based on endocrine and the nervous system, which are at the basis of many diseases. Environmental control and cleaning, where bio-nano things deployed in the environment, such as a natural ecosystem, would check for toxic and pollutant agents, and collaboratively transform these agents through bioremediation, e.g., bacteria employed to clean oil spills. Bio-nano things. Within the scope of the Internet of bio-nano things are defined as uniquely identifiable, basic structural and functional units that operate and interact within the biological environment, stemming from biological cells and enabled by synthetic biology and nanotechnology. Bio-nano things are expected to perform tasks and functionalities typical of the embedded computing devices in the Internet of Things, such as sensing, processing, actuation, and interaction with each other. Biological cells as the substrate of bio-nano things. A biological cell is the basic unit of life, consisting of a membrane that encloses a mixture of highly specialized molecules with defined chemical composition and function, which may also be organized into functional structures. A mapping between the components of a typical IoT embedded computing device and the elements of a cell becomes apparent if we compare electrons' propagation in semiconductors to functionally similar, although much more complex, biochemical reactions. In this context, as illustrated in Figure 1, some examples are as follows. And here's Figure 1. Elements of a biological cell and components of a typical Internet of Things device. We have the control unit, which contains the embedded software of the device, would correspond to the genetic instructions densely packed into the cells, DNA molecules, which encode protein structures, the cell's quote-unquote data units, and regulatory sequences, similar to the software conditional expressions. Here we have the memory unit, which contains the values of the embedded system's data, would correspond to the chemical content of the cytoplasm, i.e., the interior of the cell, comprised of molecules synthesized by the cell as a result of DNA instructions and other molecules or structures, e.g., vesicles, exchanged with the external environment. Then we have the processing unit, which executes the software instructions and manages memory and peripherals, would correspond to the molecular machinery that, from the DNA molecules, through the so-called transcription and translation, generates protein molecules with instruction-dependent types and concentrations. Then we have the power unit, which supplies the energy to maintain the electrical currents in the embedded system circuits, would correspond to the reservoir in the cell of the ATP molecule, which is synthesized by the cell from energy supplied from the external environment in various forms, and provides the energy necessary for the cell's biochemical reactions to take place. The transceivers, which allow the embedded systems to exchange information, would correspond to the specific chains of chemical reactions, i.e., signaling pathways through which cells exchange information-bearing molecules. Sensing and actuation, which allow embedded systems to acquire data and interact with the environment, would correspond to the capability of a cell to chemically recognize external molecules or physical stimuli, e.g., light or mechanical stress, and to change the chemical characteristics of the environment or mechanically interact through moving elements, such as flagella, pili, or cilia. As always, I will leave the links in the description for those who want to keep reading.